Hi, everybody. Welcome to the seminar. I'm going maskless for a minute. I'm sorry, but I can't read without my glasses, and I can't have my glasses on with my mask. So this will just be a brief moment. I'm here to introduce today's seminar speaker, Cody Sheik, from Large Lakes Observatory in the Department of Biology. Cody did his bachelor's and PhD both at University of Oklahoma, got his PhD in 2011, and then moved to Ann Arbor as a postdoc, where he was for several years. And then we convinced him to leave Ann Arbor and come here to UMD, and he started here in 2015. Cody's interests are broad, but they revolve around the diversity and functioning of microbes and microbial communities. Cody's worked in a variety of interesting and exotic locales: deep ocean, deep earth, large lakes. He has funding from almost every place I've ever heard of that gives funding to scientific investigators, NSF, EPA, USGS, and others, where he often provides really key pieces to larger projects, pieces that those larger projects really truly need. In the time that he's been here at UMD, 2 graduate students have finished. Did I get that right? And he has 5 others by my count. He's mentored numerous undergraduates. Maybe some of you are here today. And to date, he's published more than 30 research articles in journals and book chapters and the like. Cody's field of geomicrobiology is a study of how diverse microbes form an interface between the living and the non-living world. And that's what he's going to talk to us today about in his discussion about superior cyanobacteria. So Cody, it's all yours.
Can everybody hear me in the back there? All right, great. Thank you, Bob, for that introduction. I really appreciate it. And thank you all for being here. It's really great to see everybody's face and being in person again. And so it's really awesome to have you all here. So today's a bit of a departure from the normal things I like to think about on a day-to-day basis. And that's really thinking about microorganisms surviving in awkward environments, or maybe extreme environments, I guess I should say. And maybe not even like surviving, but thriving. So we think about organisms all the time living in rock. And this is rock that's really old. It's about 1.8 billion years. It's rock that's in northern Minnesota. If you're walking along up there, you might see an outcrop or something like that of it. And we also think about organisms living in sediments and how they cycle nutrients, mainly elements like sulfur and carbon and nitrogen. And so this is a bit of a departure because I'm coming up to the light. So most of the stuff has always been in the dark. That's where I like to hide, I guess. But today I'm going to talk about cyanobacteria. And this is a story that's been developing in the lab for the last several years. And I'm really excited about it. It's kind of a new adventure for me. And it seems to have like sucked me up somehow. Maybe kicking and screaming at first, but now I think I'm going to ride that current along for a while. And so with that, today I'm going to tell you if this thing works. There, don't work. Oh, sorry. There we go. Today I'm going to tell you 3 little stories that have been developing, and they're all going to sort of like maybe interweave. At least they interweave in my brain. And so the first story is really going to tell you about this organism. It's a cyanobacteria. It's called Dolichospermanii. And this organism is the primary bloom-forming cyanobacteria that's been happening in the Apostle Islands. So if everybody knows Lake Superior, we're kind of down here by my thumb. I guess I should say, and Apostles will be right over here. And so this organism, since about 2012, we've been noticing more blooms. They've been more frequent and a little bit more intense. And so we recovered a genome from that 2018 bloom. And I'd like to tell you a little bit about that organism and why I think it's a little bit special and why it can survive and thrive in Lake Superior and other Great Lakes as well. And so that's going to feed into this idea of this Dolichospermum through time. And on this second story, what I'm really going to do is try to hammer out whether or not this organism is an invasive species. And so we can think of an invasive species coming from these lower lakes, maybe arriving on the ballast waters of these big ship tanks, and arriving here in Lake Superior and then just taking hold, okay? And so I'm really going to address that question on this third act, or the second act. And then on the third act, what I wanted to really do is kind of zoom out and really think about emergent properties of microorganisms. So these microorganisms are super tiny, but they can have a dramatic impact on the biogeochemical cycles of even these large lakes. And here I'm going to focus on nitrogen fixation because this is an often overlooked part of the nitrogen cycle for various reasons. But one I think that we should really be addressing as we move forward with future climate scenarios across the Great Lakes and thinking about nitrogen budgets in general. And so hopefully I can convince you at the very end of that. So with that, kick back, enjoy yourselves, maybe grab another cup of coffee or a cookie, and let's get going here. And so you may have heard, may not have heard, about Lake Superior's growing cyanobacterial problem. Starting about 2012, over near the Apostle Islands, we started noticing these blooms occurring. And this made headlines both locally through like Wisconsin Public Radio, but also nationally. So the New York Times have covered this, and here in the last week, we actually saw the Chicago Tribune cover this. This was the front page, talking about these blooms in Lake Superior. And I think it's because these blooms in Lake Superior, we like to think about this thing as a pristine lake. Like if you compare it to Lake Erie, right? Has anybody ever been to Lake Erie? Yeah, it's kind of a, it's its own thing, right? We'll just call it that. And so when we think about Lake Erie and these Great Lakes, Lake Superior always stands out. It's like cold, it's crystal clear most of the time, right? And people want to come here. Tourists like spend weeks up here and just enjoying it, right? Being in this interface with the outdoors. And so I think that's one reason why this has really garnered a lot of attention here recently, is mainly because it can't happen up here, right? We have to think about this thing, and it can't. This is our tourist spot. This is where we always go. These things can't be happening. But it turns out they are. And we're really interested in this because from a public health standpoint, these things are extremely dangerous. There's many cyanobacteria out there, and they produce a lot of toxins. And some of these toxins target us. Some of them target zooplankton or amoeba, these grazers out in the water column. But some of them are just very broad. And so when we have these blooms, we're really worried about the public health and this public interface with these things. And so this microcystin is one you may have heard of. This is what shut down the Toledo water system and caused them millions of dollars to bring it back online here just a few years ago. This is microcystin. There's several variants of it, something like 130, maybe 150 different variants of this toxin. And it's really important. And so we spend a lot of time and money trying to measure this across lakes, not only in like the Great Lakes, but also in lakes like Peltier, which is down by Minneapolis. And this thing is crazy. It's green most of the summer. It's a nasty lake, but yet people have $1 million homes on it, right? And they're fishing in it every day. Ugh. But we also have to worry about other sorts of toxins. And so we don't have to worry about just one. There's multiple ones of these. There's Saxotoxin. You may have heard of it, called paralytic shellfish toxin. This is what happens if you eat a bad shellfish. We also have these things called anatoxins, which are these very fast death factors. And so these are both neurotoxins, and they can really do some damage to you if you're exposed to enough concentration of it. There's also new emerging ones. This one is killing bald eagles in Arkansas, right? And this is from a new cyanobacteria that's primarily down the south. And it's a brominated compound, and that's what makes this one unique, is these bromines on here. It makes it so toxic. And so we're continually finding new toxins. We're still continually finding new cyanobacteria, despite the fact that, I don't know, this may sound scandalous to other microbiologists or something like that, but that's my little one in the back. Thanks for the encouragement, hon. We're continually finding new cyanobacteria, despite the fact that cyanobacteria are one of the most studied groups of microbes on the planet, probably next to E. coli. Like, we have so much study on these things, yet we have so much to learn about them. And that's what makes them exciting for me. Okay, so let's think about the cyanobacterial bloom. I keep saying that they keep being frequent, right? So 2012, 2015, 2018 was the largest bloom that we saw. And they've all been happening here around Cornucopia Beach. And this is one of the views on that 2018 bloom. You can see tourists hanging out here, jet skiing, having fun. But these blooms are quite different than, say, Lake Erie blooms. Lake Erie blooms, basically you get your calendar out and be like, oh, April's happening, we're gonna have some blooms. And if you like watch this, it's gonna happen all the way out into the fall. And so we know a lot about Lake Erie blooms, mainly because we can study them so thoroughly throughout the year. Lake Superior is different. These blooms don't last very long. If you're lucky, they last maybe a day, maybe 2 days. In this case, in the 2018 bloom, it was like 3 days that we got access to it. And so part of actually studying these things, and I like to call them ephemeral blooms because you see them and they're gone. Unless you have eyes on the water instead of boots on the ground, you may never see these things, right? And Bob could probably tell us a lot about the history of the blooms around all of Lake Superior, because these things have been happening infrequently for like the last 60 years, right? But if you're not there on that specific day, you may never see it. And if you don't have a water, like a water bottle or something like that, you may never be able to sample it, okay? And so that's what makes these things so intriguing and so hard to study and so hard to understand, okay? But that 2018 bloom was special because it lasted a very long time. It was very big. And two, we had so many researchers come in and actually be able to sample this thing. And so Bob's group was able to get out there the day after the bloom started initiating this would be August 9th. And so they were out there on the 10th sampling from inside here. And I'll orient you over here. So Duluth would be over here, right? And so we're talking about the Apostle Islands up there, but we're looking at Cornucopia Beach, just a little bit south and west of there, okay? And so here on Cornucopia Beach, it was a really nice chalk green sort of cyanobacterial bloom. Crazy, like this thing is crystal clear most of the time. And so there's also other blooms that could have potentially been happening right around that same time. This is down by Wisconsin Point. And so you can see these spikes in chlorophyll that are coming up on the water column there. And so my group, or I say my group, our group, that was Andy Bramberger and Chris Philstrip from the NRI, they were actually out on the Blue Heron doing something completely different. But they got the call and said, hey, there's a bloom happening. And so they beat feet, or boated feet, I don't know. They headed over there as fast as they possibly could and got over there on that third day. So that'd be the 11th. And they were actually able to sample from 5 different spots for us and pull water. They kept seeing these surface scums everywhere. This is very, very common for these types of blooms. But they also were doing net tows and seeing very dense phytoplankton. And whenever I actually look inside those phytoplankton pools, we were seeing this organism. And so these are colonies here, and these are more of like at the cellular level. And so if we actually zoom back out a little bit, it looks a lot like this. So this is what they were pulling from Cornucopia Beach on the 9th. And this was identified as Dolichospermanii. In here, you can also, if you squint every once in a while, you can see these specialized cells that are called Heterocysts. And these organisms are fixing nitrogen within these Heterocysts. And so just keep that in the back of your brain for now. And so we were recovering an organism that looked very similar to it. And so Andy and Chris brought the water back, and they said, here you go, Cody, have fun. And so my lab, in my lab, we use cultivation-independent approaches many of the times to really try to get at the genome of these organisms. And so we use these DNA-based approaches to really think about these organisms. And when we do that, we basically take this water, we filter it down and concentrate the biomass. We extract DNA from that, and we send it off to a sequencer. And what we get are these little tiny reads. They're like 150 bases long. And we have to assemble these things back into something that we can actually understand and use. And so a microbial genome for these cyanobacteria typically range in somewhere around 2 megabases, all the way up to about 5 megabases in some cases. And so we have to take these 150 reads and like make them really, really big, right? But we can use these advanced algorithms and bioinformatic tools to assemble this stuff together and make sense of all these like little tiny reads. And what we get out at the very end of that are these genomes that we can taxonomically identify to the species level, okay? And so here from our miasma of sequences, we can bin out these different genomes, and then we can look at their genes on there and describe their metabolic function, right? So a protein coding gene will have a specific function. But importantly, we can look at the evolutionary history of that organism, right? So we can think about how the genome has evolved over time and diverged from other classes. We can also look at global distributions. We have really great public databases out there where we can actually go and say, how similar is my organism to all these other ones that have been deposited in this database, okay? And so when we do that, we can get this really great idea of its global distribution, potentially. But importantly, for toxin analysis and thinking about these toxic blooms, we can look for these toxin production genes. These are very well characterized in most of those toxins I just showed you. And we can think about these secondary metabolite genes that they're producing. Okay, so this was our prime suspect. We already knew this going into this. So this actually makes it really easy for us when we're trying to think about who's the culprit here. And so we already knew who we were looking for after we got all this data. And so I started going through the Excel sheet and saying, okay, what cyanos do we have in our samples? It became really easy, almost too easy, I think. We came up with like 3 cyanobacterial genomes. 2 of them were these little tiny, like what we call picocyanos. And these things are distributed all across the lakes, all across the ocean. And right now, I think we have something like 70 or 80 genomes in the lab of these picocyanos from Lake Superior. So those organisms, I can just like, we're done with these. These are just normal background flora, right? And so I went down to that 3rd one and said, okay, who am I looking at? And lo and behold, it was the one we wanted. It was this Dolichospermanii genome, okay? And so this is just showing you like the phylogenomic tree where we use about 60 proteins to really get a robust phylogeny of these. And you can see some of these different clades. It doesn't really matter about the names right now, but I want to focus on this green one. And if we blow up this green one, I want to point out all of these fall within one species. So what we're looking at in this green box is basically one species of cyanobacteria, and that's the Lemmermanii, okay? And so what I'm showing you, though, are individual genomes. So these are genotypes within there. And so we can also refer these to these as strains in microbiology. These are different strains of the same organism, okay? And I want to highlight this one up here in this blue box, because that's where our genome was, this SB001, Superior Bloom number 1. I'm not very good at naming things. And so we have our Superior Bloom one up here. And what I want to point out here is that we have this other clade that's down here and this little intermediate clade. All these ones down here are coming from Europe, like either from Finland or the UK, and they're distributed all across lakes and whatnot across Europe. And what you'll notice here is this big star is denoting which ones can actually produce microcystin, okay? And so when I was doing some initial blasting, I kept hitting this Anabaena 90, and I got real excited and also real kind of like, oh no, oh no. So I got excited as a researcher, right? Because I'm like, this is like the first microcystin producing organism to be found on Lake Superior, at least to my knowledge at that time. And I was like, oh man, that's exciting. And then I started being like, oh no, this is really potentially has big ramifications for tourism, for all these other things in this region, right? And so I got real bummed out a little bit. And I think that's true for a lot of cyanobacteria researchers. There's this duality where you're finding really cool research and then you're like, oh, but public health-wise, I don't know. And so, yeah, I'm always kind of trying to dodge back and forth on it. But what's interesting about this clade is that every one of them is actually able to produce this anabaenopeptin. And this anabaenopeptin looks like this down here, and it's got a lot of different structures. I think it's almost like this microcystin, sorry. It has like another 134 different congeners, and it's all made up of amino acids, and they just kind of smash them all together to make these big molecules. And it has an unknown human toxicity to it. There's a lot of like confounding literature out there, but all of them can make it. And I want to point out was that this peptide was actually found inside the bloom. So Todd Miller actually found this in the bloom itself. So this is a nice, really one-to-one confirmation that we're finding these genes in our organism, and we're actually seeing the toxin out in the lake, which is kind of cool. I don't know, but let's key in on this bloom, this blue box up here. So these genomes are all coming from North America, and so this one here, this MDT, that one's coming from Washington State. And these two up here are fun. These are actually coming from the Great Lakes. So we have one here, this AL09 is from Lake Ontario, and this one down here is from Lake Erie. And so we're finding the same organism, right, or just different genotypes distributed across the lake. And that's exciting to think about and some of the repercussions of these. I tried to get these cultures, so these were actually cultured at one point, and turns out there was a faulty incubator, and they burned up. So these cultures have actually been lost now since this actually was published in 2018. And it's very unfortunate. And so we've been trying to get this SB001 back into the culture from Lake Superior, and we're having some good results off of it. But looking at this, I was like, okay, why do we have the same genotype or a genotype of the same species spread across the lakes, right? And I started thinking about this a little bit and trying to say, how is this organism able to survive, right? Here we have like really high nutrient concentrations, especially in phosphorus. This is Lake Erie. This is like if you're in the summertime flying into Detroit. This is like one of my favorite things to do when you fly into Detroit, is actually see Lake Erie blooming. It's weird, I know. But you have high phosphorus, we have very low phosphorus in Lake Superior, right? And so one of the things I was trying to battle back and forth is, how is this organism actually blooming, right? How can it survive in both of these styles and environments? It makes sense that it can survive in Lake Erie. There's lots of nutrients around. And they've actually been seeing these Dolichospermanii-like organisms for since the 50s. And so it's no surprise that it's down there, right? This is just a big soup of fun for a microorganism. But Lake Superior is a little bit different. And so I started thinking about this a little bit more. And if they say that, the eyes are the window to the soul, right, for humans, hold on, just hang on with me. The transporter systems that microorganisms have are the view into the soul of the organism. I know, it's a stretch. It's fine. And so these transporters, though, if we think about it, they are the ones that are bringing nutrients into the cell. So if I need to get phosphorus into my cell, I have to have a transporter. If I need to get nitrogen in, I have to have a transporter. And so I started looking at these transporters. And I want to highlight these ones that are here in these two boxes, because these are keying in on phosphorus and iron and manganese. And what's fun about these is that these use ATP to really bring in these nutrients. So things that are at very low concentration outside the cell, they're trying to suck it in as much as they possibly can, right? And so this organism's using ATP to do this. Turns out it's a phototroph, so it doesn't really matter if it's using energy. It's got this light source that's constantly replenishing its ATP and other things, right? And so it can expend that energy to bring this into the cell. And what's fun about this is it actually has genes that actually make this phosphorus mineral, what we call polyphosphates down here. It uses more ATP. But what it does is that it stores this phosphate for long-term growth in this mineral form, and it can suck on it and eat it. Maybe not suck on it, but it can dissolve it away and eat it whenever it needs nutrients, okay? What's also cool about this organism is that it also has the ability to store iron. And so it can chelate this iron and hold it inside the cell until it needs it. It turns out things like photosystems really need a lot of iron. There's a lot of other things like nitrogenases that need a lot of iron. And so this organism can store that iron whenever it needs it, right? And I don't think they have any manganese ones, but it'd be interesting to see if they have any capability of storing manganese. But anyway, let's keep trucking here. But also, this thing is just like accumulating everything, I think. We have things for PHBs. These are big, long alkanes, like just carbon storage. And then we also have nitrogen storage. These things are called cyanophycins, and they all feed into this ammonia pathway here. And so they can store nitrogen for long-term usage. And so I got real excited about these transporters, and I was like, man, this is crazy. But then I started looking at all the nitrogen transporters. I'm like, this is a nitrogen fixer. It can make its own nitrogen if it wants to. But then it can also soak up all this other sorts of nitrogen. And so we have things for nitrate, nitrite. We have things for ammonia. And then if we go over the organic side of things, we have urea. We have things for amino acid uptake. And so I really think this organism is just like a survivalist. It's ready. It's got its bunker, and it's just waiting to hear for somebody to be like, get down in the bunker, right? Because it's got everything already loaded. It's ready to go. And so it's a hoarder and a survivalist, I think, in general. And I think that's one reason why it's able to survive in these really stoichiometrically different lakes, say with Erie and Lake Superior. Okay, so we've seen that it's in Lake Erie and Lake Ontario, but I think one of the big questions is it elsewhere in Lake Superior? So, is it just happening on the Apostle Islands and this is the only place that we're seeing it, or is it elsewhere? And so my graduate student, Jake Callahan, was analyzing some data from the 2016 CSMI study. This is through the EPA. And he has all these different stations that we've gone out to, both in the spring and the summer. We have multiple depths, multiple filter sizes, blah, 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 blah. We have a lot of data, turns out. And what we're doing is using a 16S rRNA gene. And so this is a really good taxonomic record of microorganisms. And it allows us to go out and look at the distribution across an environment just by using this one gene. And so he was looking at this stuff and just trying to think about broad patterns of diversity that we see in Lake Superior. And I told him, I was like, hey, let's pull out the cyanos and just take a look at this. And so when he did, he graphed this up. This was a huge panel graph. But so I was just highlighting a couple here. And so what we have here is that in the springtime, we have some cyanos that are always hanging out. But we also see some that are blooming. They're growing in the summertime. This makes sense, that they want to key in on the right temperature. So they're growing. And we have these Aphanizomenon, Dolichospermum, and Planktothrix. And I was like, oh, well, hey, we're looking for a Dolichospermum. Maybe this is the Dolichospermum we want. And so we started looking at this, and I was like, oh, wait, you know what? I have a 16S gene that I pulled from the bloom, right? Let's go in and actually see if we can do a one-to-one comparison using a program called BLAST. It basically allows us to give a similarity of our sequence to another sequence. And so when he started using BLAST on this to like really suss this out, turns out it wasn't this one. It was actually this Aphanizomenon right here that we were looking at. And one of the fun things about cyanobacteria is that the taxonomy is awful. And it is like just across the board, both by microscopy and molecular tools. And they don't sync. Like there's very little syncage right now between these different data sets. And it can be frustrating, but it's also exciting. And so this thing was actually hiding in our data set because it was misclassified, right? And so when we started looking at this group, I said, okay, let's pull this out and just kind of look at it and its distribution across the lake. We actually found multiple variants of this one. So all of these would fall within one species, like if we use our traditional species language. But what we're looking at are single nucleotide variants. So basically, this one may have an A at position 250. This one may have a T at position 250, and it's conserved, right? And so we can say that this is a very conserved variant. And so when we look at this, we see all these different variants. Some of them are occurring in rare areas, you know, they're maybe hitting in some localities very sparsely. In our data set, we have these nearshore environments. We also have offshore environments. But I want to highlight this one right here, or I did highlight this one right here, this ASV 721. You may notice that it's found in every station we went and sampled at, okay? Some of them are different abundances, so the larger the circle here, the more abundant it is in our data set. But we're finding it everywhere we go. And I think that's really telling and really exciting to think about that we have all this variation within the lake, and some of these are actually coming up quite routinely within our data sets, okay? Okay, so to kind of summarize that part, and that's gonna set us up for the next one, is that this organism is a hoarder, it fixes nitrogen, and it's distributed across the lake in the summertime, okay? I think those are the three big things that we need to keep up. This is just a view from the lake. I always like dangling off the side while we're out, and I'm probably gonna lose my phone one day, but it'll sink to the bottom. But maybe I'll core it out one day, 10 years from now. Anyway, so let's really hammer out this idea. So we have this organism, it's distributed across the lake, it's distributed across Lake Erie, Ontario. Most likely it's in Michigan and Huron. Like I would bet money on this, that we'd find it whenever we go and sample it. So is it an invasive? And I think this is a really big question that looms over a lot of cyanos, and as we think about climate change and things like that, right? Are these things moving around and being transported in different regions and then blooming, okay? So is it an invasive? I think our sediments might be able to tell us some more. So when I first came to UMD, I was really excited by sediments. I still am. I will talk your ear off about sediments in Lake Superior. But we needed some data. So we went out in 2015 on a cruise, and we went to these 6 sampling stations, all the way up by Isle Royal. We did some out here in the middle of the lake. We went out to the Apostle Islands. This one's over by Castle Danger, and we went one right by the harbor here. We call it St. Louis Station. And so when we went out, we took a multi-core. In this instrument, we basically drop it off the back, let gravity do its thing, and it goes and plunges down into the sediment, and it can pull out a full core that's somewhere in the range of maybe 30 to maybe 50 centimeters, depending on how much weight we smash on the top of this thing. And so we spend a lot of time on the back deck or down below in these big hoods that we take on board so we can be very clean about it. And basically, we're going layer by layer by layer by layer, 1 centimeter at a time, sometimes half a centimeter at a time. But essentially what we're doing is we're going back in time. So the deeper we go in these cores, the farther back into time we're going, all right? And so I had these data sets hanging around and you know, always like trying to like figure out what's happening in these things. And I was like, you know what? Why don't we try and take that 16S that we found from the bloom and try to mine our data out to see if it's actually there. And so when I used that same BLAST program, I came up with this, another set of ASVs, another set of variants that are found within the sediment. And some are very sparse, found like only one of our stations. Some of them are found at all of our stations, okay? And so I want to highlight just 2 here. Let's focus in on just these 2. I should also point out, these are also abundance. So the bigger the circle, the more abundant it is. But these are also extremely low abundances. I want to point that out. And so these would actually be considered rare fractions of our data set, okay? Or rare members. So let's look at these 2 here. So this one, this 1797, is really being quite prominent and being deposited at SU19 and SU17, which is right here and right here. These are 2 of our lower sedimentation rate stations. This one's like out in the middle. Like some people hate going to this station. I love going to this station. It's like you can't see land anymore. You're just floating off. Like you don't get cell reception. It's great. It's like you can just like lay out there on the back deck maybe. I don't know. Might get seasick. But these 2 are very low sedimentation rate areas. But what you can see is that starting off here in these younger sediments here at the very top and working our way down to these older sediments, you can see it routinely showing up in this data. So it's being laid down and being deposited on the bottom of the lake, right? And it's being buried over time. And it's a nice contrast, I think, to this one right here, this 3246, where you see it actually being more prominent and prevalent here at the AI West. And so our AI West is right here. Turns out this is actually very much in line with where these blooms have actually been happening. It's offshore, yeah, but it's on that very same side of where we're seeing these blooms from the apostles happen. And so we're actually seeing this ASV show up quite regularly. And if you squint and just kind of be like, suspend disbelief maybe for a little bit, you might be able to say that some of these are showing patterns of blooms, right? Where you see some of these show up very infrequently, but they show up really abundantly, right? So you can imagine if you had a bloom, it's being like a lot of biomass and then it's being deposited down, right? But we should take that with a grain of salt or a grain of sediment, I don't know. But one thing I really want to point out is fun, is to do these back of the envelope calculations. I love doing these because we're scientists and we like to think about things. And so I did the sedimentation rates. And so I wanted to know how old some of these things were, right? Like what would this 10 centimeters correspond to? And so I took sedimentation rates from Evans et al. We weren't able to do these cores and date them. It's very costly. And so we were doing this on a shoestring budget, but Evans went out and did like 20 different stations across the lake. And so I basically went and took their data and said, all right, let's calculate the median, mean, a minimum rate of sedimentation, and the maximum rate of sedimentation, because that's going to get us stations like SU17, and it's also going to get us stations like St. Louis or this AI West, where they have high sedimentation rates. And so what's fun is that if we go out here to these, like a station like AI West, where it may be a little bit higher sedimentation rate, we may be looking at 10 centimeters, something like 30 years, okay? That's not bad. That could be like riding the currents on some of these boats. But we have to remember we have a bunch down core too, right? And we also have these very low sedimentation rate areas like SU17. And so if we go out there and actually find this thing at 10 centimeters, we're looking more on the average of like maybe 100 to maybe 300 years that thing has been buried, okay? And so to me, that seems that this thing's been here for quite a long time, right? And so I think for some of these variants that we're seeing, we can probably not label them as invasives. They invaded at some point, right? Mainly because this place was all ice like 10,000 years ago. So at some point, they've been migrating up, but it's hard to tell at this point. But they've been here for a while, okay? And so just as a sanity check, I was like, all right, Jake, we should probably check this out and make sure that we're checking to see if we're seeing like the same thing happening in the water column that we're seeing being buried. And it turns out we are. Like, these are the same ASVs that are present in 2016 that are probably still there now, are also ones that have been buried there for a long time. And so I think this was just a nice one-to-one thing for us to be like, oh yeah, we're seeing real things here. And some of you may be like, Cody, how do you know that DNA is being preserved, right? I think that's a big one. Like, is this just BS and you're pulling our leg, or is this stuff real? And so I had a grad student, I kind of forgot about this for a while, and I'm sitting there, I was like, I have this data somewhere on my hard drive. And so I had an undergraduate student, Austin Saylor, and he really needed a project, and he needed like a credit to graduate. And I was like, all right, you come in the lab in the summer, we're gonna do something fun. And so I had just gotten done reading this paper from one of my colleagues, John Kirkpatrick, in geology. And he was studying sediments out in the Pacific Ocean that were 100,000 years old. And so when he was going down to these 100,000-year-old sediments, he was still finding chloroplast DNA being preserved down there. And so he was actually able to reconstruct somewhat what the phytoplankton community looked like 100,000 years ago from these sediments, right? And so I was like, oh, okay, cool. I think we should do this. Let's try to do this in Lake Superior and see if we can differentiate some of these pools of DNA. And so Austin did a whole bunch of different DNA extractions to really try to get at this. But in blue here, I want to point out, this is extracellular DNA. This is stuff from lice cells that's either adhered to like a clay matrix, so DNA has a charge, and so it can adhere to like clays and whatnot and intercalculate in between those things. It can also just hang out like on silica shells of diatoms and whatnot. But what you'll see is that, it starts off up here at the top of the core. There's a lot of variation within this. This is like a very detrital rich area of the sediment, so it makes sense. But as we started going down core, it stabilizes and stays pretty steady all the way down to 20 centimeters. And then this brown box here is all the stuff that's within membranes. So it's membrane bound. It's like it's an intact cell, essentially. They may be snoozing, right? But it's an intact cell that's taken a nap, right? And so if we look at this intracellular, we see this nice relationship. Karen Lloyd showed very similar relationships with cell counts in the ocean when she was here. And so you see these things decreasing in size, that fraction pool size decreasing as we go down core as well. And so that's exciting. So that's telling me that these organisms at some point are dying, that DNA is being turned over and it's being adhered to the sediment, and some portion of that is being preserved. And so we actually sent this off for sequencing when Austin was around, and we got these sequencing results back, and I started mining them out to see whether or not there was actually cyanobacteria actually being in these sorts of different fractions. And it turns out we found a bunch of different cyanobacteria. These are all the just different genera that we actually recovered from this core. And don't worry about this as just abundances. These are all very rare sequences. But you can see that we're getting a lot of these picocyanos being laid down all the way throughout the core. And at some point, the actual community starts to change. Like once you get down to that 20 centimeter area, we start seeing different organisms showing up and persisting over time. So this really was to me said, hey, this is real. I think we are seeing real things happening here. And so just to kind of wrap this up here, these sediments, I think, are a really great record. And I think they're going to be a really great tool for us as we move forward. We're already proposing to do some of this work on Lake Erie and Lake Huron. And so I think this may be able to give us some clues of past climate, and thinking about how microorganisms migrate across these lakes and some of these maybe interesting interconnections that we may not be seeing. Okay, all right, so Act 3, I'm gonna wrap this all up here. I know, get some time going. So I wanna revisit this nitrogen fixation thing. So organisms, I mentioned like Dolichospermanii, have this ability to fix nitrogen. And so when I first got here, it was really this graph. This is what started this whole fascination with nitrogen fixation. As an undergrad, I tried to cultivate diazotrophs. These are organisms that eat nitrogen gas and make ammonia, right? And I did this in class, and they were like, hey, you probably don't want to do that. It's going to take a really long time for them to grow. It did take a very long time for them to grow, but they finally grew. And so I guess maybe it stems to like undergrad time. And I just kind of put that in the back of my mind. But this graph was above the water cooler, and Bob had a poster of this. And so every day I'd come in the yellow, get my glass of water, and I'd stare at this graph. And I'd do this multiple times a day, multiple days a week, right? And just kind of stare at it and be like, wow, what does this mean? And it finally dawned on me. I might be slow, I don't know. But it finally dawned on me. So what we're looking at here is nitrate concentrations on the y-axis, and this is in micromoles per liter. And down here, we're looking at time. So since they started measuring this back in the 40s, around the 40s, we've seen nitrate increasing. And it turns out it's actually 12 micromoles per liter that it's actually accumulated. And so some of you in chemistry may know, like trying to make like reagents or something like that, that if you've had too much cups of coffee, trying to make a 12 micromolar concentration of solution or whatever, if you jiggle your hand too much, you're going to beat it to scale for a very long time. Turns out it's not that much nitrogen. But then it dawned on me, Lake Superior is huge, right? So it's like 10 to the 16 liters of water in Lake Superior. So doing these size calculations, I was sitting at my desk and I started giggling and I was like, oh, this is so fun. Just got a piece of paper out and like I've got my calculator and I get this final number and I'm like, what does this even mean? Like I couldn't even fathom how big this number was that I was looking at. And so I put it on a scale of something, of the biggest animal I'd actually seen physically in my life. And that was an elephant. And so when I did this and I converted over this nitrate into elephant sizes, it's a mass elephant equivalency. This is the African elephant. It's about 13,000 pounds if you want to know, on average. It turns out it's about 1.5 million elephants' weight has accumulated in Lake Superior since like 1940. And that blew my mind. Then it also blew my mind. I was like, how big are these things? I know they're quite long. So you could actually line these things up, tip to tail, and go to the moon if you wanted to, and just walk for a casual stroll along its back and almost get all the way back. And that's exciting to think about these sorts of scalar sorts of things that we never like really think about. And so it turns out this is a lot of nitrate. And so this was also in that paper by Finlay in 2007, but it was also on that poster. And so I started looking at this. This is the isotopes of nitrate. So here we have 18 oxygen that's present in that nitrate, and we also have our 15N that's present in that nitrate. And so this gives us an idea of like kind of sources of where these things are coming from. And so here you can see precipitation it's depleted in that 15N, much like the nitrate is, but it's also way off on the oxygen. But streams and rivers is more enriched in 15, right? So it's a little bit heavier, and its oxygen is getting close to that. Turns out this nitrate that's in Lake Superior has an oxygen signal that is very much on line with like the normal background water, right? So the oxygen has to be coming from Lake Superior water that's getting added to this nitrate. And this nitrogen is also depleted. So what's happening here is that organisms, like microorganisms, love to fractionate nitrogen and other elements. So it's always going for like the lightest version of this. And so we also see this thing being depleted. And so when I started thinking microbially about this, there's really only two ways to get to nitrate. So nitrate's right here. One, and it all kind of goes on the same path. So we can either recycle organic nitrogen that's coming in or being produced. So things like amino acids are being turned over, continually being made and putting out ammonia. Or we can have nitrogen fixation. And nitrogen fixation can go in and feed into ammonia, and then it can go all the way up through nitrification or Comamox to get back to nitrate. And so thinking about this, we've been thinking more about organic nitrogen here recently. But I really keyed in on the nitrogen fixation because I thought we could actually measure that really easily in the lab because we had this old GC that still ran. These things are workhorses. And so sometimes it just takes like a right student to do some of this too. I'll put that and bring that in. But anyway, so getting back to this nitrogen fixation. So I started looking around to see if there was nitrogen fixation rates for across the lake. Not only Superior, but anything across the Great Lakes. And it turns out there's none. Like, it's very sparse. Like, there's probably data hanging on people's hard drives that they've collected over the years, but for published work, it's actually very scant. And some of the last work that's been done on Lake Superior was in 1973 here by Mag and Burris. And what blew me away when I was reading their paper was that they went in with just this giant mesh, like micron mesh, a 64 micron mesh. Just for reference, I use a 3 micron and a 0.22 micron to catch the organisms. And I know that there's still organisms that are passing through this 0.22, like viruses and other ultra tiny bacteria. And so it blew me away that they were actually looking at this to concentrate it and actually measure this nitrogen fixation rates. And so after I got done reading this, I was like, my head was I thought it was gonna explode. And I was like, they're missing like all these organisms that we know are very small and hanging out by themselves and are doing nitrogen fixation. And so I really wanted to go back and actually do this. And it took a while to find a student, but finally Kayla came into the lab, what was that, 2018? And I was like, you should do this. And she was like, I don't want to do that. I want to do cyanobacterial blooms. And I was like, but cyanobacteria fix nitrogen. And she was like, not doing it. Somehow in like the preceding month, I think she came back and said, I want to do this now. And I was like, yeah, you do. That's a great idea. We should do that. I'm glad you thought of it. And I don't think that's how it went, but anyway. And so we got her on a ship. So she did spend a lot of time in Lake Superior trying to like work through the methods, get the GC back up and running, testing different columns. But when we finally had it down, we finally got her on a ship. And this is the RV Lake Guardian, the research vessel Lake Guardian. It's operated through the EPA. And she went down and started off over here in Milwaukee and did basically the lower lakes all in about 2 or 2.5 weeks. I think it about killed her. And really, I was like, oh, I hope she stays in science. She's going to be doing great. But this data set is the first real comprehensive look at nitrogen fixation rates across the lakes. And we did it not only through surface waters, which is what Mag and Burris did. They basically leaned over the boat and was like, I think this is probably good. But we know that a lot of microorganisms don't live in that top half of the water column. The light is too intense for them, right? And so we went and did a depth, chose multiple depths here, multiple stations all across the Great Lakes. So you can see our map here. Basically, the size indicates how much nitrogen fixation is happening, and we also have them color-coded here as well. And so here you can just see, just spatially across these lakes, that there's quite variable, especially if you get down in here to Lake Erie and Lake Huron. And so that's exciting to see, that we're having quite a bit of spatial variation amongst these lakes. And again, this is like, it only took maybe like a day and a half or 2 days to actually do this entire Lake Huron here, right? And so spatially, like temporally, these things probably aren't changing very much, right? We're probably capturing a really good snapshot across this lake at one good time, okay? And so what's really cool is that we started seeing these depth-dependent nitrogen fixation rates happening all across our samples. Some of them are more significant than others. Like Lake Erie it's weird, it's shallow, and so there's actually not that much difference between bottom water and then surface water on nitrogen fixation rates, which is really interesting. Just because it's not significant doesn't mean it's not interesting. But I want to point out here this little spot you may have noticed down here in Lake Erie. So this was a bloom that Kayla and them came across, and when they looked at it on the microscope on the ship, she saw this, and I think she texted this to me. She's like, "We got Dolichospermanii," and I was like super happy about this one station that they had, and all amongst this sea of other cyanobacteria. And it's been known that this Dolichospermanii has been hanging out in Lake Erie for a long time, and it blooms a lot in the central basin. But finding it amongst this Microcystis was really fun. And lo and behold, when she was actually doing these nitrogen fixation rates, those are the highest rates she was finding, right? And so there was a lot of potential in demand at that one station at that time for growth. Anyway, but I also want to point out this, is that in Lake Michigan and Lake Superior, their nitrogen fixation rates are very similar. And I think some of this is that Lake Michigan has been changing chemically ever since the invasion of mussels, right? And so they've seen multiple mussel invasions. Same thing with Lake Huron, but this lake is really changing chemically and really quickly. And so I don't know, maybe Michigan, Bob probably has some ideas about this, but maybe Michigan will be like Lake Superior with its nitrogen problem quite soon. But I should point out, we published this earlier this year in Journal of Great Lakes Research, if anybody's interested. And so this was the other thing that really stood out to me in her data set when we started going through this data. There's been this long-held sort of belief in water ecology, is that if there's nitrogen around, nitrogen fixation should be very minimal. And this is what I think has prevented people from actually thinking about it and actually going out and measuring it in many ecosystems, is that if they're measuring nitrate or ammonia out there, they're going to be like, we don't need to measure nitrogen fixation. It's not occurring. Or it's occurring so minimally that we don't have to worry about it. And so when we started graphing out this data, and the EPA doesn't actually measure ammonia, unfortunately. There's a lot of problems with measuring ammonia. And so just using nitrate here off this data, you can see that lakes like Michigan and lakes like Ontario here are kind of holding to this dogma. Like we see decreasing slopes on these, right? As we increase nitrogen, we're seeing the nitrogen fixation rate decrease. But for lakes like Erie here, and also lakes like Huron, this isn't like holding true at this time point that we sampled at. And this is actually very similar to what's happening in like the Northern Atlantic, which is very oligotrophic area. You actually see extremely high nitrogen fixation rates happening there. And so in some of these areas, despite there being nitrogen around, potentially the nitrogen demand is so high that nitrogen fixation just doesn't matter. Like it doesn't care. It's still going to start doing things. But I also think about these non-cyanobacterial nitrogen-fixing organisms and thinking about them being distributed across the water column. So if we think about the surface, we had really high rates at the surface, and we had these really low rates sort of down towards the bottom. We have two actually populations. We have these phototrophs that are doing it, but we also have these diazotrophs that don't care about the light. And they don't really care about temperature because they're down at 4 degrees if they're in Lake Superior throughout the year, right? And so this really got me thinking again, and I got my pencil out and started doing these more back of the envelope calculations that I love to do. And so I started thinking about, okay, what's the contribution annually of these nitrogen fixers? And so basically I took the low end, so our minimum, calculated a mean, and I also calculated and looked at the max rate for each lake and said, okay, if we scale this to a full year, what do we see? And you know, this for the max rates is again light dependent, not going to happen year round. But what's fun is thinking about those organisms that are just like trolling the bottom. Like they don't really care. Like it's 4 degrees to maybe like 8 degrees year round. It's dark. These lakes are deep. And so at the very minimum, I think yearly, we're getting tons of nitrogen being put into the system from these organisms that are just doing their thing. Like they don't care, they're just doing their thing. And so you can see like for like Erie, you know, like 10 tons of nitrogen. That's new nitrogen being put in the system yearly. What's fun about Lake Superior, right? It's so big. It's like, you know, what, like 70, 80 tons of nitrogen are coming into the lake potentially, just if we use the basal sort of our minimum rates. And in the summertime, that could spike up. So we might actually see something like this, you know, if we're thinking about it as a holistic sort of approach. And so this gets me really excited to start working on this and doing more focused studies and more long-term studies. And I've been talking with people that have been studying nitrogen fixation in Lake Erie, and they're excited about doing this too. And so we're going to start trying to think more about nitrogen budgets of these Great Lakes, just based on some of these graphs that we did in Kayla's work. And so again, I think this nitrogen fixation is something we really have to start thinking about, especially for nitrogen budgets, when we start thinking about the long-term health of these lakes and where they're ultimately going to end up and what the ultimate community is going to look like 10, 15, 20 years down the road, okay? All right, so I've told you a lot about a lot of stuff today, right? And so I thought I'd just kind of step back and like really think about what this actually means on both like a global and sort of a regional scale as we finish out these last few minutes here. So blooms like Microcystis blooms are increasing worldwide. And so here you can just see, if you see a red dot here, this basically means that they're increasing in frequency, in intensity, and sometimes toxicity. And so some lakes I've just pulled out from this paper here by Ho et al. Really show like the response of Lake Erie. It was doing really well. They were managing this lake, and the concentrations, changes in fertilizer and things like that really started improving Lake Erie. But at some point, it just started like, you know what, guys? I want to go back to where the good old days, right? Where I was blooming all the time. And so it has actually been back on the increase in that lake. Lake Huron has kind of like been like, I don't know if I want to, maybe I won't, I don't know. And lakes like Michigan are just kind of laying flat. And so the whole gist of this paper was that there's multiple environmental drivers that could be driving all these blooms. And there's not really just one sort of target catch-all driver that's happening. And I should point out, this is just for Microcystis. We can do this for every sort of genre or every species of cyanobacteria and say, okay, they're all probably going to follow similar patterns here. All right. And so one of these big drivers that we've long known about for cyanobacteria is temperature. And unfortunately, Lake Superior is one of the fastest warming lakes on Earth. So thinking about temperate lakes, lakes that have ice in it. So this was a really great paper by O'Reilly. It came out, and you can really see this trend here that we're increasing. But Jay Austin here in the physics department and also at the Large Lakes Observatory was doing some long-term monitoring and just going back through old data sets of Lake Superior. And this is just air temperature that you can see. So it's mean air temperature that's happening. And there's a really direct relationship between air temperature and surface water temperature. And you can also see that ice cover is decreasing throughout time as well. And so these lakes are warming up faster. And because of that, we're not getting as much ice. All right, and so we also have these nutrients that we've also been known to be a very strong driver. This is ELA Lake 226. This was classic work by Schindler, where they basically were like, let's go dump a bunch of like phosphorus and nitrogen in these lakes and just see what happens. And like, this is like pristine. Anybody that's been up to the ELA before, Bob can say this too, it's beautiful. Like, it is gorgeous up there. And it's so isolated and remote, it's fun to be up there. And they're basically like just polluting these giant lakes just for science. And it's great, right? And we get to like all this knowledge for it. But we've long known that like phosphorus is a really strong driver of these blooms. And what's fun is that these things became nitrogen limited, and we see those like Anabaena and those nitrogen fixing species actually hanging out in here, fixing nitrogen, and actually driving these blooms. And so we actually have big test beds, right? This is Lake Erie. This is also Lake of the Woods that we've also been studying this summer. And Lake of the Woods is weird. This is an extremely long-lived bloom this year that they've had, but this, you can also time your watch to this one as well. And so where am I going with this? Well, it seems like our Duluth is really blowing up nationally, not just for like these blooms, but people want to move here, right? And so we keep seeing like these articles being like, Duluth's the best place to live for climate, right? And like, come here, we got mountain biking, we got all this other stuff, live here, right? And so part of this, when I think about long-term timing, is that Duluth at one point is going to get more populated. Like that's just like the northern is going to, the Arrowhead is going to get more populous. And because of that, we're going to be changing land use, right? And so we could be seeing more nutrients like phosphorus and all these other runoffs, because people like to, throw nitrogen fertilizer and other sorts of things down on their lawns. So you have this like beautiful lawn. It's inevitable that we're going to get some more nutrients coming into the system just because of like people influx. And so, I don't know. I'm at odds here with this, right? Because one, it's great for me for a scientist because we can actually study these things more routinely, right? And keep funding for my lab. But two, it's like, I hate to see this ecosystem sort of change right before my eyes. And I would like to like end this on a great note. And maybe I will, I don't know, I'll turn it around. But unfortunately, I don't think that blooms are gonna go away anytime soon. I think they're here to stay. And we saw this summer where we had blooms happening on Park Point Beach. We also had blooms happening inside the harbor here. And unfortunately, like for this Barker's Island bloom, we actually had microcystins being detected, and we also had saxotoxins being detected. This is a prime area on the Wisconsin side for swimming in this little cove. And so I think we have to be more wary about moving forward about sampling and stuff. But I think one of the great things, it's goodish news, right? I think it's good news, is that we have a collection of researchers in the Twin Ports area that are now like primed and ready to go and sample these blooms and really fundamentally think about these blooms and why that's happening and why they're driving and what's going on with them. And so I think this is really going to start getting at some of these long-term models and start thinking about, okay, my lab at least is thinking about how can we integrate this genomic data? Like when we start thinking about these different strains and these different genotypes, are they moving in? Are they coming in from like invasions? Are they coming from the land, right? And so we can actually trace these things out potentially and say, okay, this one is being sourced from maybe like deep in the bay, right? The St. Louis River. Or maybe this one's coming from offshore. And so using these genomic tools, I think we can actually start to like generate new models to really start thinking about what's driving these blooms and what to look for, right? If it's a hot, dry year, yeah, we may need to start like watching this area really intensely and keeping swimmers at bay. But we're also doing some like just classic cultivation all the time because we want to get a big database of these. And unfortunately, there's going to be new players, I think, moving into the system. We already saw some new players up here. But this is a culture that we have growing in the lab right now. I'm generally calling this, it's a new genus. I'm calling it Siskowitzensis, meaning it derives from Siskowit Bay. And this organism is interesting. It's a nitrogen fixer. You can see these heterocysts here, but it's also producing a very novel toxin. And we don't actually know what this toxin is right now. And when I'm working with collaborator Todd Miller, and some other people at Milwaukee to really understand what this toxin is. And what they've been doing is putting this to zebrafish models and just trying to see what happens. And this toxin is interesting because like at 40 parts per billion, it causes death within most of these zebrafish when they expose these embryos to them. And so I think we really have to be keen about understanding these blooms and these organisms right from a fundamental standpoint. And the more we sample, the more we'll know, and the more we can predict and things like that. So if I was going to lead on a good note, it's that. We're trying our hardest to be out there in the water and trying to get these things as much as possible to make it's safe to drink water or boat or fish and things like that in Lake Superior. So I've tried to acknowledge as many people as I can throughout the talk here. And I just want to acknowledge our funding sources through UMD that help some of the sequencing for the bloom. NRI also provided some money. I get some money from NSF for some other projects right now, and EPA for our CSMI work. And so with that, I appreciate your time, and thank you, and I'll take any questions you might have.
All right, thanks, Cody, for the update. On the Sheik lab, where all the science is superior.
I'd like to think so.
We're pressed for time. If anyone needs to get up and go, totally understand. And we'll go through the formal questions kind of quickly, but I'm sure Cody would be happy to stay behind. And
sure, definitely.
Longer. So if you need to excuse yourself, please go ahead. We'll start with questions from the undergraduates.
About, you're kind of ahead of the curve with all this data, like really treating out and staying on top of it. Do you think with like the potential of blooming population-wise, there could be potential for collaboration between your labs or someone in agriculture or trying to prevent that?
Yeah, I think definitely so. So the question is like the collaboration network that happens with some of this and thinking about agriculture in general. Yes, very much so. I think, especially like in the southern part of the state where you have that really agriculture interface, people are really like keen on understanding blooms and stuff down there because they are quite toxic. A lot of these ones down in the southern one, you saw with like Peltier and things like that. And so we've actually been studying some lakes with different land use change around them. So agriculture lakes versus populous lakes versus stuff that's in timber, and trying to understand these blooms as well that are happening there. So yeah, definitely the coordination network is expanding greatly. Yeah.
Does nutrient redistribution and concentration associated with past blooms facilitate blooms at all?
Yes. So in places like Lake of the Woods, right, we know that there's a sediment component to that, and you get this recharge effect where phosphorus is being released back from organic matter, degrading over time. It's a big story with sulfur and things like that too. But you do see this recharge event where these organisms are going down to the bottom of the lake during the wintertime, and they're just kind of hanging out. And that's a very nutrient-rich zone down there. And so it kind of primes them, especially in Erie, right around the Maumee River. You actually get the microcystis accumulating where all those nutrients are coming in. And you actually see growth happening early in the spring, and then at some point they just kind of pop up to the surface. And so, yes, past blooms definitely is a big thing.
Undergrad's still welcome, but let's also open up to graduate students.
What are some of your factors or like things you're looking for when you're picking a site for like your sediment blooms?
So some of those stations were long-time stations that the EPA has used. So SU-17, and 19 are those really long-lived ones. Isle Royal one, we just wanted to find something near Isle Royal, and we're like, this looks good. And we knew that it's from some other work on Lake Superior from our colleagues at the LLO, that there were some really good sediments in there. And so some of it is like we kind of have a good idea already of what's happening. Some of it's just blind luck. Like the St. Louis core, that one's a really hard one to get because there's a lot of sand, but we somehow managed to pull a core from there. And so some of it is blind luck. Some of it is that we actually go out and you can actually look at the sediment depth and the characteristics of it and be like, okay, this is a good place to core. And so we've done that a little bit. And Bob's been doing that too.
More questions from undergrads or grad students? Faculty, anyone else?
Dovetails nicely with Dylan's question. So when thinking about what might trigger these things, obviously it's complicated, but is there any relationship between, so I immediately think of the depth at which those things are resting and how much pressure is on them or how far up they must go. And also with temperature changes, you think about changing currents or changing things like that. Do you think depth or currents are playing a role?
Yes, and I think there's also places we have different sources. So some of them are potentially keying in on these river outlets, like the one from Siskiwit Bay and that one. And so these big rainfall events are delivering nutrients constantly into these bays, right? And so you could have explosion of growth just because you have more phosphorus coming in, right? But I think also with those ASVs showing it, those ones that are distributed across the lake, it's showing us that there's probably stuff that's riding right along in that area where we may not actually be sampling. So the EPA goes out and samples at 20, 15, 5, or 10 and 5. And that's it. They don't do anything in between that. So 5 meters is big, right? And so you could have these zones where some of these cyanobacteria are hanging out that we're not actually sampling routinely from. And so I think that each one of these organisms is going to have their own sort of concentration of light they like, nutrients, and things like that. And so very much, we need a better study, essentially.
What role does atmospheric nitrogen deposition play in the system?
So just by what we saw there, Bob did actually a nice, in 2007 also, showing a big nitrogen budget of it. And if I'm trying to remember, it's like 3 to 5%.
I can't remember what I had for breakfast.
It's substantial. It's substantial, yeah. But in the comparison of other pools, it's actually quite pretty small. Like organic nitrogen inputs coming from like riverine is actually much larger than the actual deposition. I do remember that part from the table.
That was, I can add to that in terms of context, that was the early explanation for that nitrate rise. People looked at it and said, well, that's just NOx pollution, we're done. And then the isotope studies and other things indicated, no, the lake is making its own nitrogen somehow. And that's part of the excitement of looking at new fixation, nitrogen fixation. So, and then the other aspect of that is nitrate or other forms of nitrogen being produced are also being consumed, and it's that net balance that somehow has shifted. So, yeah. Anyone else?
So this is kind of a crazy question, and I'm just going to ask it, and then I'll explain why I'm asking. How long does a cyanobacteria live? And so the reason that I'm asking that is we have an organism that is apparently investing a lot of energy in these transporters and hoarding all this stuff, and that just seems counterintuitive for a short-lived organism that I imagine a bacteria, or a cyanobacteria must be.
Yep.
So
it's a good question. So these things live eternally. And I say that in jest, but also they produce these actinides. So these are spore-like resting structures. And so essentially what we're probably seeing in the sediment are these actinides hanging out and just waiting, right? If that sediment gets like redistributed or something like that, they can come back up and start to bloom again. And so that genotype can hang out for a long time and persist in the environment. But I think when it's just actually growing, we'd have to look at a death rate. I know Bob's student Kate has been doing some modeling studies of the blooms itself, and there's an internal death rate within that. And so you could actually probably tune that and like try to figure out, just do some modeling studies. But it's probably maybe like a week or two, like a normal cell would probably last. You know, it's all based on nutrients, and like at some point they're going to get enough like just junk hanging out in the cell that they're just going to be like, I can't handle this. But also viruses. And so that's one key I haven't like talked about in here, but we're also trying to put the virus to the actual cyanobacteria with these data sets as well, because we can mine those out. And so viral is actually a huge component of the recycling. And so if you add that component in, like it's probably less than a week that each cell is going to be hanging out. Yeah.
Thanks, everybody.
Thank you, everyone.